One minute, 1 minute. Hello, hello. Is it okay, guys, now? Yes, now it's okay. Okay. Hope everyone have gone through the yesterday's class. Uh, Many of the students are asking like where exactly we can get the PDF. Guys, after completing the general physiology, all the general physiology topics will be uploaded in one PDF format. Okay, just wait for the next few days. We will upload the entire general physiology in one single book format. Now, let's continue with the other aspects, other topics in the general physiology. In yesterday's class, what we have discussed about the cell membrane and all the related topics. Now, in this class, we'll be mainly concentrating on the membrane transport and related topics, and wherever possible, I will try to integrate it with the pathology and the clinicals. Okay, so in this class, as you guys are not exam going batches, I will be mainly concentrating on the basics. Okay, I will be emphasizing on the basics, guys. Now, having said that, let's start our today's topic. That's the membrane transport. Membrane transport. Now, my question for you is, how many types of membrane transport do you know? So, the membrane transport is mainly of two different types. Membrane transport is of mainly two different types. First one is active transport, and second one is passive transport. Okay, active transport and passive transport. Now, what is the exact difference between the active transport and passive transport? So even in your intermediate, you have already studied these things, right? In your class 11th and 12th also, you know about these things. So active transport requires ATP for the movement of the substances, for the transport of the substances. ATP is required for active transport, but in passive transport, there is no need of ATP, no ATP molecules. There is no need of energy. This is the MCQ. Okay, if you don't require any energy for the transport of substances, that type of transport is called as a passive transport. And in active transport, you need to use ATP for the transporting of the substance. Now, having said that, first let's start our topic of passive transport. Passive transport. Okay, our first topic is passive transport. Now, if I use the word passive transport, then itself it should come to your mind that there is no need of any E. Now, in passive transport, first let's see what are the subtypes. Okay, passive transport is just a heading. Under this, there are a lot of subtypes. The first subtype is simple diffusion. Simple diffusion. Now, I'm asking you, does simple diffusion require any ATP for the movement of substances? No, simple diffusion does not require any energy. First of all, what exactly is this simple diffusion? Okay, what exactly is this simple diffusion? Now, here the substances are going to move from high concentration to low concentration. Okay, especially here I'm talking about the gases. A gas, for example, oxygen, oxygen will move from high concentration to low concentration naturally. If you take oxygen here, which is having high concentrated oxygen here, and in the other area where the concentration of oxygen is less, automatically by nature, by default itself, oxygen will move from high concentration or high partial pressures to low concentration. So this type of movement does not require any ATP. It does not require any ATP. Simple concentration gradient is enough. Concentration gradient means from high concentration to low concentration. Gases will move. Such a type of movement is called as a simple diffusion. Okay. Example here I am giving you is gaseous exchange. Gaseous exchange. Now, here let me show you. This is alveolus. Okay, this is the alveolus in your lungs. Alveolus are the places where you have oxygen, and down to the alveolus, blood vessels are present. Which blood vessels are present, guys? This blood vessel is a pulmonary capillary. Here is a pulmonary capillary. You are breathing, right? Oxygen is going to your lungs. Oxygen is going into your alveolus. and at the same time your heart is pumping the blood to the lungs. So blood is coming to the lungs in the pulmonary capillaries. Now right now the blood is in the pulmonary capillaries. Now see, in the pulmonary capillaries, oxygen concentration is more or less. In the in the capillaries, oxygen concentration is less. Here in the alveolus, oxygen concentration is more. So automatically, oxygen will cross these layers and oxygen will enter into the pulmonary capillaries. So there is a transport. There is transport happening. Transport from the alveolus into the pulmonary capillary. So this type of transport, which is following the concentration gradient without use of any ATP. So such a type of transport is called as simple diffusion. Okay. Now, at the same time, in your blood, the blood which is coming to your lungs for the purpose of oxygenation. So this is a deoxygenated blood, right? Deoxygenated blood is coming to your lungs. Deoxygenated blood contains carbon dioxide. So carbon dioxide concentration or carbon dioxide partial pressure is more inside the pulmonary capillaries, and the carbon dioxide concentration in the alveolus is very less. So what happens? Automatically carbon dioxide will move from the pulmonary capillary into the alveolus. So this is also an example of simple diffusion. So if someone comes and asks you gases exchange, this transport, the transport of gases occurs by active process or passive process? Occurs by passive process. Which type of passive process? Simple diffusion, not the facilitated diffusion. It's a simple diffusion. Now, as I am discussing about the simple diffusion, first let's see, diffusion depends on diffusion of gases or diffusion of substances. It depends on certain factors which is explained by, diffusion is explained by a law called as Fick's law. Okay, Fick's law. So what Fick's law says is the diffusion of any particular thing depends on concentration gradient, it also depends on surface area. Don't worry, I will explain you in detail. Okay, surface area, and also diffusion is directly proportional to lipid solubility. Okay, and concentration is indirectly proportional to thickness. Okay, so Fick's law explains us diffusion depends on which factors. For example, let's see the first factor, diffusion, sir. It's directly proportional to the concentration gradient. If more is the concentration gradient, more diffusion will happen. What exactly I mean by is, for example, let's take two alveolus here. Okay, two alveolus here and two pulmonary capillaries. Okay, now this is A and this is B. Now here in the first A alveolus, the partial pressure or the concentration of oxygen is 100, let's say, and the concentration of oxygen in the pulmonary capillary, for example, let's take it as 0. Okay, this is a hypothetical condition. Just let's take something like this. In the second alveolus, the partial pressure is again 100 here. Partial pressure of oxygen is 100. In the pulmonary capillary, the partial pressure here is 90. Okay, in the pulmonary capillary, it is 90. Now tell me, in both the cases, in both the conditions, will diffusion happen or not? Just tell me. Definitely diffusion will happen, sir. Why? Because see, 100 is high and 0 is less. So diffusion will happen, no doubt. Even in the second condition, here 100 is greater than 90, right? 100 is greater than 90. So here also diffusion will happen. But where concentration gradient is more? Sir, in the first condition, concentration gradient is more. 100, 0. More difference is there. More concentration gradient is there. So more diffusion will happen. Much diffusion will happen. But in the second condition, here 100 is there, here 90 is there. Partial pressure difference is just only 10. Diffusion will happen, but less. So in which condition more diffusion will happen? In the condition number A. So that's what Fick's principle is saying us. So diffusion is directly proportional to the concentration gradient. More concentration gradient, more diffusion will happen. Less concentration gradient, less diffusion will happen. Okay, let's take the second thing, that's the surface area. Diffusion depends on the surface area. Now again, let's see two more scenarios here. This is the first alveolus and this is the second alveolus. something like this. Now, alveolus number 1, alveolus number 2, A and B. Now, in which case the diffusion will be more? So, in A, alveolus A, there is more surface area. There is more surface area. As there is more surface area, there is more possibility for the diffusion to happen. Okay, there is more possibility for the diffusion to happen. In the case number B, there is very less surface area when compared to A. The B have less surface area, so automatically diffusion here will decrease. So that's what. If more is the surface area, more will be the diffusion. And very important point is the lipid solubility. Guys, any substance which is lipid soluble will be having more diffusion. Especially here when I am talking about the gases. Remember, for your entire life, all gases, oxygen, carbon dioxide, whatever, gases are lipid soluble. They can easily cross through the cell membranes. For example, in yesterday's class we have seen cell membranes, right? Cell membranes which are made up of phospholipids. Okay, cell membranes like this. All the gases, oxygen, carbon dioxide, nitric oxide, whatever. For example, here is the oxygen. Oxygen will easily cross. So what I am saying is, gases are any substances which are lipid soluble. Those substances will have higher diffusion rates. Okay. Now, after this, let's talk about diffusion relation with the thickness. Now, let me show you again two alveolus. This is alveolus number 1. This is the alveolus number 2. Now, you have to tell me, sir, here is the pulmonary capillary. This is the also the pulmonary capillary. Now see, because of some reason, in case number 1, in this case, the alveolar basement membrane, okay, the alveolar basement membrane is so much thickened. Now, this condition is called as pulmonary fibrosis. Okay, this condition is called as a pulmonary fibrosis. So what happened in pulmonary fibrosis? Name itself is pulmonary fibrosis. The pulmonary basement membrane or the alveolar basement membrane, it got thickened, it got fibrotic. Now in the second condition, it's absolutely normal, physiologic. Now tell me, in which condition diffusion will be more and in which condition diffusion will be less? So you can think logically and say, here oxygen is there, here also oxygen is there. Here oxygen can be easily diffused. But in case number 1, oxygen cannot cross. Even if it cross, the diffusion will be less. So from this we can clearly say, if the thickness of the membrane increases, means if you increase the thickness of a membrane, automatically diffusion is going to be decreased. So that's what Fick's law is telling us. Diffusion is indirectly proportional to thickness. If someone comes to you and asks you, diffusion is explained by Fick's law. Diffusion is directly proportional to concentration gradient, surface area, as well as the lipid solubility. Diffusion is indirectly proportional to the thickness. Now, in the exam, these kind of questions will come. See, that two direct questions which will come in your exam is condition number. First condition is emphysema. They will ask you. And second condition, that is pulmonary fibrosis. So, in emphysema, what happens? And in pulmonary fibrosis, what happens? Now, if you don't know, okay, let me ask you. Okay, if any second year students or third year students, if you are really there, guys, what exactly is emphysema? Can you tell me? Emphysema is due to deficiency in which enzyme? Anyone? Come on, guys. Emphysema is due to. I'm checking your comments. I have started checking the comments. Come on, Ahmed, Chandra, Hubs. Anyone? Emphysema. Do you know this pulmonary condition called as emphysema? Okay, whenever you go to these movies, like the advertisement will be coming, right? Don't smoke cigarettes. If you smoke cigarettes, you will be suffering with that emphysema. Okay, what exactly is emphysema? Excellent, iconic. You are true. Alpha 1 antitrypsin deficiency. Medico, you are also true. See, emphysema is a condition due to deficiency of alpha 1 antitrypsin. So, alpha 1 antitrypsin deficiency causes emphysema. Now, tell me, in emphysema condition, what happened to the diffusion? Our topic. What happens to the diffusion? Diffusion increases or diffusion decreases? First of all, let me show you what happens. See, this is how a normal alveolus will be. This is how a normal healthy alveolus will be. If you have emphysema, alveolus will become something like this. Alveolus will become something like this. Now this is emphysema. This is normal. Now, what is the difference, sir? Guys, are you able to appreciate this alveolar septas? Okay, this is the extra membrane, guys. This is the extra membrane where gases exchange will happen. There is extra membrane. But here in emphysema, all that extra membrane is lost. Means the surface area is lost. In emphysema, what happened to surface area? Surface area is decreased. If surface area is decreased, what happened to diffusion? The diffusion also decreases. So remember, for your entire life, what happened to diffusion in emphysema? Diffusion decreases. No doubt, diffusion decreases. Now tell me, in pulmonary fibrosis, what happens? Sir, in pulmonary fibrosis, okay, this is how the pulmonary fibrosis will look like. The entire alveolus, the alveolar basement membrane will become more fibrotic, Now what happens? Now do you think the gases will exchange properly? No, gases exchange is not happening properly. So in pulmonary fibrosis, what happened to diffusion? Diffusion decreases. Okay, both emphysema and pulmonary fibrosis, diffusion decreases. So simple diffusion, some important related points completed. Now let's start our second topic, that is the facilitated diffusion. Facilitated diffusion. Now here my question is, facilitated diffusion, is it an example of active transport or is it an example of passive transport? It's an example of, definitely it's an example of passive transport, no doubt. Okay, do you need any ATP in facilitated diffusion? No sir, no need of any ATP. Then what exactly is the difference between facilitated diffusion and simple diffusion? See, even facilitated diffusion also, the transport of substances are happening from high concentration to low concentration, no doubt. Okay, movement of substances are happening from high concentration to low concentration. But important point here, you especially need some carrier proteins. Carrier proteins are involved for the movement of the substances. Substances are moving from high concentration to low concentration, no doubt. But here, what do you need? You need a carrier protein mediated transport. So that direct MCQ in your exam is carrier protein mediated transport is facilitated diffusion. Now comes the question. So can you give any examples of such a facilitated diffusion? Facilitated diffusion. Can you give me the examples? Any examples? From your MBBS study? It's the GLUTs, glucose transporters. Glucose transporters and aquaporins. Now let's discuss a few important points about the glucose transporters and aquaporins. See, normally, here let me show you adipose tissue. Okay, this is an adipocyte. Okay, this is an adipocyte. We know all adipocytes are the stores for fats. Now, here is a blood vessel. This is a blood vessel. Okay, in the blood vessel, what do you have? The glucose is there. Now, this adipocyte have to take the glucose. Now, glucose molecule, it is trying to enter the adipocyte. Now, do you think this glucose molecule can enter into adipocyte normally? No. Here, see, glucose is high in blood. No doubt, glucose concentration high in blood. Glucose levels in adipocyte is less. So, glucose is moving from high concentration to low concentration. Concentration gradient is there. But what is the problem? The glucose molecule cannot cross this cell membrane. Now, what should happen? See, what happens is, I am going little bit in detail. Normally in your blood, who is there? Insulin is there. Now, I am asking you. Now, insulin, it will act on the adipocyte. Now, my question to you, if you are really good, just try to answer it. Now, insulin will act on which receptor? Can you tell me? There is a receptor for insulin. The receptor name, can anyone tell you? The receptor name is called as a tyrosine kinase receptor. So insulin use which type of receptor? Tyrosine kinase receptor. The moment insulin comes and binds to this tyrosine kinase receptor, do you know what happens? Now this cell will plant certain transporters here. This transporter is now present. Before it's not there. Now it's coming. Now this transporter is called as glucose transporter. Okay, this transporter is called as GLUT, glucose transporter type 4 MCQ. Type 4 is MCQ. Okay, glucose transporter type 4 is present on adipocytes and skeletal muscles also. Okay, now only this glucose molecule will enter into the adipocyte, and here the glucose is now stored in the form of the fats. Okay, now after this, where exactly these aquaporins are present? Where exactly these aquaporins are present, guys? Do you know where exactly these aquaporins are present? If you know this, try to just put it in the chat box. Where do these aquaporins are present? Aquaporin molecules are present in, not glut5. Someone is saying glut5. Satish, it's not glut5. Glut5 is used for fructose transporters. Okay, now yes Medico, you are true. Aquaporins are present in nephrons. So the important MCQs which you should know for your exam is, see, now I am showing you a nephron in a easy way. See, this is the Bowman's capsule. Later you will be having proximal convoluted tubule. After that you will be having the descending limb of loop of Henle, hairpin turn, ascending limb of loop of Henle, thin segment. After that you will be having thick segment. Later on you will be having distal convoluted tubule and collecting tubules. Okay, now see, we all know when water, okay, when water is filtered here in the Bowman's capsule, now this water, it's coming where? It's coming to the proximal convoluted tubule. Now after this same water, okay, the same water, the plasma I should say, okay, the plasma is getting filtered. Now this plasma is coming into the proximal convoluted tubule. Plasma is nothing but the water, right? The plasma is coming into the Bowman's capsule, proximal convoluted tubule, and now this same plasma is coming, it's filtered, and now these water molecules are coming to the descending limb of loop of Henle. This is descending limb of loop of Henle. Now what should happen in the nephron? So you need to reabsorb the water. Okay, water should be reabsorbed. Now important point is, here you are having these carrier proteins. These carrier proteins are present in the descending limb of loop of Henle. What is the name of this carrier proteins? These carrier proteins are called as aquaporin 1 channels. MCQ. Aquaporin 1 channels are present on the descending limb of loop of Henle. Now what these aquaporins are doing? Now these aquaporins, now they will try to, they will help to reabsorb the water. So this is the way by which water is moving from the nephron into the interstitium, and from the interstitium this water will be again taken back into the systemic circulation. Okay, so water reabsorption happens with the help of aquaporins. Which type of aquaporin? Aquaporin 1. Now if you are really good, especially if you are exam going batches, now where do aquaporin 2 present? Where do aquaporin 2 present? The aquaporin 2 is present on collecting ducts. In your nephrons, there is a region called as collecting duct. Okay, there is a region called as collecting duct. Now, see, this is something important, especially for the exam going batches. Whenever you have antidiuretic hormone in your body, see, whenever you have antidiuretic hormone in your body, now this antidiuretic hormone will act on collecting ducts. Okay, collecting ducts. Now what happens? Now this antidiuretic hormone will help in implantation of this aquaporin 2 channels. Now whenever there is aquaporin 2, water reabsorption happens. Now water is being reabsorbed. So two direct important MCQs. Aquaporin 2 channels are present on collecting ducts. Okay. Now second MCQ, if you really if you are really good, just try to answer it. Sir, antidiuretic hormone, two important MCQs are there. Antidiuretic hormone is produced from, can you tell me? Antidiuretic hormone, it is produced from which part of the body? Antidiuretic hormone is produced from which part of the body? Come on, Sweta, Medico, Shoaib, Satish. Antidiuretic hormone, also called as vasopressin. It's produced from which part of the body? Excellent, excellent. It's the posterior pituitary. Okay? Wrong. It's not the posterior pituitary, guys. Never ever say antidiuretic hormone is produced from the posterior pituitary. Produced from hypothalamus. It's produced from hypothalamus. See, Sweta, Medico, Bruce, you're all saying it's produced from posterior pituitary. No, it's produced from the hypothalamus. From the hypothalamus, it's produced, and now it comes down and stored in the posterior pituitary. Okay, in the posterior pituitary is stored in the vesicles. The antidiuretic hormone is stored in the posterior pituitary in the vesicles. So, first important MCQ, antidiuretic hormone is produced from hypothalamus. Stored where? In the posterior pituitary. Okay, posterior pituitary. Now, this antidiuretic hormone is acting on which area? Collecting ducts. So, what is the receptor, sir? See, this antidiuretic hormone, it will come and act on a receptor. Okay, you need to have a receptor. Now, what is the name of this receptor? Can you tell me? See, previously I have shown you insulin is acting on a receptor. What is that? What is this kind of receptor? It's a tyrosine kinase receptor. Insulin receptor is tyrosine kinase receptor. No doubt. Next question here is antidiuretic hormone. It's acting on the collecting ducts. Now, what is the name of this receptor, sir? Can you tell me? V2 receptor. Yes, it's a G protein coupled receptor, no doubt. G protein, GPCR, G protein coupled receptor. But main in exam point of view, what you should answer is it's V2 receptor, sir. V2 receptor. Okay, antidiuretic hormone acts on V2 receptor, no doubt. Okay. Now, at the end of the day, let's come back to our original topic. Our topic is facilitated diffusion. So, what exactly is facilitated diffusion, sir? Transport of substances are happening from high concentration to low concentration, no doubt. But what do you need? You need to have a carrier protein. Examples of carrier proteins are aquaporins, glucose transporters. Okay, they are examples of carrier proteins. Okay. Now, after this, let's talk about the third type of passive transport. The third type. What is that? Osmosis. Okay, osmosis. Okay. Here, what exactly is osmosis? Here also same. It's an example of passive transport. There is no need of ATP. No ATP. But what is happening here? In osmosis, we are specifically talking about water movement. Okay, water movement. From where water is moving from, yes, high concentration to low concentration again. High concentration to low concentration. But what is the difference, sir? Here water is moving via a semi-permeable membrane. Here important point is there is a membrane separating high concentration, low concentration, but through a semi-permeable membrane. This is a semi-permeable membrane. Who is moving from high concentration to low concentration? Water or solvents. Okay. Now, in which type of, the question that will come you, in which type of transport semi-permeable membrane is involved? Primary active transport, secondary active transport, simple diffusion, facilitated diffusion, vesicular transport, or osmosis. In osmosis, semi-permeable membrane is involved. Now, for the movement of water, you need to require a force. A force is required. Don't worry, it's going to be very easy. But just for now, see, the required force in osmosis is called as osmotic pressure. Okay. Now, what exactly is this osmotic pressure, sir? Now, in order to understand that, I should show you a simple experiment. This is beaker number A. I am taking a beaker and beaker number B. Okay, beaker number A and beaker number B. Now, I am just connecting these two beakers. Okay, I am connecting these two beakers like this. Okay. Now here is a semi-permeable membrane attached. Here is a semi-permeable membrane. A semi-permeable membrane is there. This is very important. Okay, having said that, in beaker number A, I am putting water. Here is a water. And in beaker number B also, I am just putting solvent. That's the water again. Okay. Now, water will move from, it's a pure water. It's a pure water. So, water will move from beaker number A into beaker number B or beaker number B into beaker number A. There is no net transport of water, sir. There is no transport of water. But let's see, I am doing something different. Okay. Same thing. Here, please concentrate. I am again taking same beaker number A and beaker number B. So what I am doing is, in beaker number A, I am trying to add salt into it. Sodium molecules. I'm adding sodium molecules here. That's the salt. Normally, before it's a pure water, right? Pure water concentration is how much? Pure water concentration is almost zero. Okay, the osmotic pressure is almost zero. Now, right now, what I'm doing is I'm keeping more sodium here. Now, what happens? So now this water is becoming more concentrated. Now the water is becoming more concentrated. Okay, here most of the students will confuse. So I'm adding more salt. Now water is becoming more concentrated. Now tell me, in this condition, water will move from beaker number A into beaker number B or beaker number B to beaker number A. What is the net movement of water? So here, see, the water will start to move from B to A via the semi-permeable membrane. What exactly happened here? So, wherever there is sodium, water will try to go towards sodium. Remember, for your entire life, or wherever sodium goes, water will go along with the sodium. Why? Because sodium is having a potential called as a osmotic potential. Sodium is osmotically active substance. So it tries to attract the water towards itself. Okay, so water now is moving or not? Yes sir, water is moving. So transport is there. Okay, transport is there via a semi-permeable membrane. So this type of water transport through a semi-permeable membrane is called as osmosis. Okay, so this is the example of osmosis. Okay, now how to understand or how to apply this concept, sir? How to apply this concept in our real life? See, for example, I'm taking a blood vessel. Okay, let me show it in a red color. Or for, okay, let me take a cell. Okay, let me take a cell here. I'm taking a cell and I'm taking interstitial fluid here. This is the interstitial fluid. So here cell membrane, guys, remember here cell membrane is acting as a semi-permeable membrane. Okay. Now here the concept which you have to understand is, normally our blood is not a pure fluid. Our blood, okay, our blood is not a pure fluid, right? Our blood is having extracellular fluids. Blood is an example of extracellular fluid. Our blood is having an osmolarity of 300 milliosmoles. Now we should all the time maintain our blood osmolarity as 300. We have to maintain our blood osmolarity as 300. Now you can ask me, sir, why it is important? For example, if you decrease the osmolarity, what happens? If you increase the osmolarity, what will go wrong? See, for example, right now what I am doing is, now I am taking too much amount of water. So if I am taking too much amount of water, water is going to my stomach, water will be absorbed in systemic circulation. So what happened to the extracellular volume? What happened to extracellular volume? Sir, extracellular volume increases. Now water, I am diluting, I am taking extra water. If I am taking the extra water, what happens? Sir, extracellular volume increases. So from 300, see, this is extracellular fluid, right? This is all extracellular fluid. Now what happens? Now osmolarity here, normality is 300. How much it will become? Now it will come down to 200. Okay. Now osmolarity here, it is decreasing. Okay. Now you are decreasing the osmolarity. Now what happens? See, from the cell, now from the cell, water will come out. From the cell, water will come out. I will get it out. Why water is coming out, sir? See, just concentrate on the before condition. See, water will, sorry, just 1 minute, guys. I just made a small mistake. What happens? Water will try to go into the cell. Okay, now water will try to go into the cell. Okay, why? See, here, let's take them as two cells. This is a real cell. Okay, just take it as a cell. Now, all the time, water will move from low concentration here in the cells. There is no sodium or there is no solutes here. No solutes are there. Now, here, please concentrate. In the beaker number A, more solutes are there. Water is more concentrated. Water is more concentrated. Water is more concentrated when compared to beaker number B. So automatically fluid will move from here to here. Even here in the same way, whenever you drink too much amount of water, pathologically if you drink too much amount of water, what will happen? Sir, your extracellular fluids are more and more diluted. Now whenever extracellular fluids, whenever they are more and more diluted, the osmolarity will come down. Now what happened? When osmolarity is coming down, water will shift to places where there is high osmolarity. So automatically fluid will now shift into the cell. Now what happens? Cells will receive more water, cellular swelling will happen. Okay, cellular swelling will happen, cells will swell and even they will die. Okay, even they will die. Okay, so pathological drinking of water you have to avoid. Okay, now tell me, so whenever you drink too much amount of water, what happens? Extracellular volume increases. What happened to the concentration? What happened to the concentration? The fluids are becoming more and more diluted. Now from where to where water will shift? Water will shift from extracellular component into intracellular component. So cells will start to swell and even they will rupture. Now the same thing, whenever you do dehydration, whenever you are dehydrating, now what happens? This is your cells, these are your cells, and this is your extracellular fluid. This is your extracellular fluid, especially. Let's take it as blood also. Okay, let's take it. It's an extracellular fluid. Now, whenever you are dehydrating, you are not taking any water. So what happened to extracellular fluid volume? Extracellular fluid volume is now decreasing. You are losing the water. Every day you are producing the urine, right? So urine is going out. So what happens? Extracellular fluid volume is decreasing. Whenever extracellular fluid volume decreases, now it's becoming more and more concentrated, right? Okay, now here more concentration increases. The concentration, okay, the concentration of the blood or the concentration of the extracellular fluid increases, which means simple, concentration increases means just think like more and more sodium is present here, more and more sodium. Why? Because you are producing the urine, so water is being removed out of your body, water is being removed out, so sodium is getting more and more concentrated. Now what will happen? Now fluid will shift from the cells into the extracellular component. So what happens? Now cells will undergo dehydration, or cells will undergo shrinking. Okay, so I have explained to you what exactly is osmosis, and I have given you two clinical important examples. Okay, whenever you do over drinking, what will happen? Cells will well, or whenever you are undergoing dehydration, what will happen? Cells will shrink. Why? Because water will move from, or simply try to understand like this, water always move towards the sodium. Wherever there is sodium, water will go towards that side. Okay, so this type of transport is called as osmosis. Now after discussing osmosis, now let's start our next topic, that is the active transport. Okay, active transport. Now, very simple guys. So, three types of passive transport we have discussed. What are the three types of passive transport? The first type of passive transport is simple diffusion. Second type is facilitated diffusion. And third type is osmosis. Where do you need carrier protein? Facilitated diffusion. Where do you need semi-permeable membrane? In osmosis. Okay. Now, let's talk about the active transport. How many types of active transports do you know? Two types. There are two types of active transport. First one is called as the primary active transport. Okay, primary active transport and secondary active transport. Okay, primary active transport, secondary active transport. Now let's start our topic with the primary active transport. First of all, both primary active transport and secondary active transport, in both types, what do you require? We require ATP. For the movement of substances, we need ATP. So can you give me examples of primary active transport? Yes, sodium potassium ATP. Sodium potassium ATP is the sodium potassium ATPase, which is present on all your cells in your body. There are trillions of cells, right? On all these trillions of cells, there is a protein called as sodium potassium ATPase. See here in the name itself it's there, ATPase, ATP broke down is happening. With the help of this ATP broke down, who is moving? Sodium is moving, potassium is moving. So sodium and potassium are moving in and out of the cells with the help of ATP. It's an example of primary active transport. So can you give me any other examples? The first example, second example is proton pump. Remember, for your entire life, whenever you listen this word pump, pump is nothing but ATPase. Pump is nothing but primary active transport. Pump means ATP is getting broken down. Okay, so first example is sodium potassium ATPase, sodium movement and potassium movement. Proton ATPase are proton pump, H plus pump. Protons are moving with the help of ATP. And third example is calcium ATP is calcium ATPase. So here also ATP is being used and calcium is being mobilized or transported. Now my question, what should you know regarding sodium potassium ATPase for the exam? What you should know regarding proton pump for your exam, and what you should know regarding calcium ATPase for your exam, sir. First, let's start our topic with the sodium potassium ATPase. Now, where it is present? It's present on each and every cell of your body. The direct questions that will come in your exam is, see, for example, I am showing you one cell. Okay, here is sodium potassium ATPase. Okay, sodium potassium ATPase, actually it have two components, two subunits. Okay, it have two subunits. This is the first subunit and this is the second subunit. Okay, let's take it. Two subunits are there. The first subunit is called as the alpha subunit and beta subunit, alpha and beta subunits. Now, especially on the alpha subunit, MCQ, on the alpha subunit, see, this is the extracellular, right? Okay, in the extracellular domain, there is an area. Okay, and intracellularly also there is an area. What is the important, sir? See, these are the binding sites. These are exactly the binding sites. The sodium, not sodium, the potassium will come and bind here in the extracellular domain. In the extracellular domain of alpha subunit, potassium will come and bind. In the intracellular domain, sodium will come and bind. Okay, sodium will come and bind, as well as ATP. ATP will also come and bind. Sodium and ATP molecules are coming and binding to intracellular binding site of alpha subunit, and potassium is binding to extracellular domain. Now what will happen, sir? So see, now what happens is, by breaking down the ATP, by breaking down the ATP, energy is released. Now sodium is thrown out, sodium molecules are thrown out, and potassium molecules are brought into the cell. So transport is happening or not? Yes, transport is happening. Who is being transported? Sodium molecules are transported out, potassium molecules are transported in. Now you should get a question, sir, why we are calling it as active transport? Because why ATP is really utilized here? Why? Because see, already in the cell you are having high concentration of potassium. Your cells are bags of potassium. Lots and lots of potassium is there inside your cells. Already outside your cells, there is a lot of sodium present. But what this sodium potassium ATPase is doing? Now it is throwing the 3 sodium molecules at a time. It throws 3 sodium molecules out of the cell and brings 2 potassium molecules into the cell. Is it according to concentration gradient, I am asking you? No, already potassium is more inside the cell, sir, but still potassium is brought. So this is against the concentration gradient, not according to the concentration gradient. So whenever such type of movement is happening against the concentration gradient, you need to use ATP. That's why here ATP is being utilized. So in a single way we can say potassium is moving from low concentration to high concentration. Outside potassium is less, but inside potassium is more. But still, by using the ATP, potassium is moving from low concentration to high concentration. That's why you need to use the ATP. Okay. Now, second MCQ that can come in your exam is, what is the coupling ratio? COUPLING, coupling ratio. What is the coupling ratio of sodium potassium ATPase? Coupling ratio is 3:2. What exactly is this 3:2, sir? It brings, or I should say, it throws 3 sodium out of the cell. 3 sodium are being thrown out of the cell, and 2 potassium molecules are being taken into the cell. The coupling ratio is 3:2. Okay, completed. Now, other MCQs that can come in your exam. What are the functions? What are the functions of sodium, potassium, ATP, ACE? Okay, the functions which you should keep in your mind is cellular volume regulation. Cellular volume regulation. So, what exactly is this cell volume regulation? Cell volume regulation. Don't worry, it's very simple. I have already explained to you. For example, let's take this as a one cell. Okay, let's take this as a one cell. Here is who is sitting here. I am showing you the sodium potassium ATPase like this. This is the sodium potassium ATPase. Now, what this fellow is doing, sodium potassium ATPase, it's throwing the sodium out of the cell. It is bringing the potassium into the cell. Okay. Now, for example, guys, I'm asking you, because of some reason you are activating the sodium potassium ATPase. You are activating it. Now, whenever you activate the sodium potassium ATPase, what will happen? It throws more and more sodium out of the cell. Now, if you activate sodium potassium ATPase, more and more sodium is coming out of the cell. If more and more sodium comes out of the cell, what happens? Water will follow the sodium. Osmosis. Water follows the sodium. So, what happened to cell volume? The cell volume decreases. So, direct question, if you activate sodium potassium ATPases, cell volume decreases. So, cell volume regulation is under the control of activity of sodium potassium ATPase. Okay. Now, other MCQ which will come in your exam. Sir, who activate the sodium potassium ATPase? The substances or the drugs which are activating sodium potassium ATPase, and the substances which are inactivating the sodium potassium ATPase. Very, very important. Now, I am asking you, so there is one hormone in your body. If that hormone is there, that hormone will directly come and activate the sodium potassium ATPase. Increases the concentration of sodium potassium ATPases on the cell. Can you tell me what is that hormone increasing the sodium potassium ATPase concentration? Can you? Come on, guys. Sodium potassium ATPase activity is increased by thyroid hormones. See, even aldosterone, some other hormones, dopamine also increase. But anyway, see, simple here. Thyroid hormones. Remember, for your entire life, what are the thyroid hormones? T3, T4. T3, T4 hormones will increase the sodium potassium ATPase activity. Okay. Now, who inhibits the sodium potassium ATPase? Can you tell me? Who inhibits sodium potassium ATPase? So the clinical importance, there is a very important clinical relation is there. I will discuss about how thyroid hormone acts on the cells. What will happen? We'll discuss in a minute. But my question right now is, who inhibits sodium potassium ATPase? Insulin? No. The answer is Digitalis. There is a drug called as Digitalis. Okay. Also, there is a one more drug called as Obein. Okay, digitalis and obein are the substances which will inhibit sodium potassium ATPase. Now let's see the clinical importance, guys. Here is a one cell. Okay, here is one cell. Okay. Now, right now I am talking about T3 and T4. T3 and T4, what are they? Thyroid hormones. Now my question for you is, the thyroid hormones are coming from the thyroid gland, no doubt they are coming from thyroid gland. Now these thyroid hormones should have to act on the cell. Now, what is the receptor present on the cell surface for thyroid hormones? On which receptors it will act? On which receptors thyroid hormones will act? Now, if you are thinking about some receptor, you are absolutely wrong. Why? Because thyroid hormone does not have any receptor on the cell surface. Now, these thyroid hormones, especially T3, the active form, the thyroid hormone will cross the cell membrane. It will enter into the cytoplasm. Okay. Now, this thyroid hormone, it will enter into the nucleus. It will enter into the nucleus. Inside the nucleus, who is sitting? Inside the nucleus, we are having DNA with the genes. Now, this T3 hormone, it will bind with its receptor. Thyroid hormones, it will bind with this receptor. When the thyroid hormone binds with its receptor, now this complex, hormone receptor complex, will activate the genes, so that transcription will happen. Later, translation will happen. Transcription, translation. Okay, translation happens in the cytoplasm. Transcription, translation happens. At the end of the day, what do we have? After transcription translation, now we are left with a protein, sir, which is called as sodium potassium ATPase. I will get it out. Sir, why we are discussing all this thing? Because see, whenever I increase thyroid hormones on your body, okay, whenever I increase thyroid hormones in your body, what happens? At the end of the day, sodium potassium ATPase concentration on the cell increases, sir. Normally, let's take something like this. Normally, there are only 3 sodium potassium ATPases on your cell. Normally. But if I increase T3, T4 hormones, if I increase the T3 hormone, what happens? So, because of transcription and translation, more sodium potassium ATPs are being produced. Now, what they will do? Now, what they will do? Just tell me, sir, each and every sodium potassium ATPs, what it will use? It will use ATP, sir. So, more ATP are being utilized, more ATP are being broken down. So, when more ATP are being broken down, more calories are being broken down. What happened to your basal metabolic rate? What happened to your basal metabolic rate? Sir, basal metabolic rate increases. The number of calories you will burn every day is called as the basal metabolic rate. For example, right now me as an adult male, I will use somewhere around 2500 to 3000 kilocalories. So that's my basal metabolic rate. Now you came to me. Now you are giving thyroid hormones. You are giving thyroxine hormone to me. Thyroid hormone. What happens to my basal metabolic rate? That thyroid hormone acts on every cell in my body. More number of sodium potassium ATPases are going to be present on the cell membrane. More ATP is being broken down. More calories are being utilized. So what happens to basal metabolic rate? Basal metabolic rate increases. So remember, till the time your exam, see, thyroid hormones increases the basal metabolic rate. That's the first question. By how? By how? By increasing the sodium potassium ATPases. Now second question, sodium potassium ATPase is an example of active transport or passive transport? It's an example of active transport, sir. That too, it's an example of primary active transport. Why we are calling it as a primary active transport? Sir, what is exactly primary active transport means? Primary active transport means direct utilization of ATP. ATP are directly utilized at the site. At the site of transport, ATP is directly utilized. Called as a primary active transport. Okay, at the site of movement, of sodium and potassium, ATP is being utilized. That's the primary active transport. Completed. Now, guys, can anyone tell me, if you are really good, if you are a second year students, you can tell me, sir, what exactly is this Digitalis? Digitalis, can you tell me what exactly is this Digitalis? What kind of drug is this Digitalis? Come on, guys. Bruce, I want you to answer. Bruce, Medico, Ayushi, anyone? Can you tell me what exactly is this drug called as Digitalis, and when we will give this drug? What kind of drug is it? Digitalis. So Digitalis is an inotropic drug. Inotropic drug. Okay, it's an inotropic drug. Yes, yes, yes. See, Amir, you are saying increases the calcium. Can you tell me how it increases the calcium? See, again I am showing you. Digitalis acts on your heart. Digitalis acts where? On your myocardium. Okay, this is your heart cell, myocardial cell. Okay, now on your myocardial cell, who is sitting? There is something called as sodium potassium ATPase. Sodium potassium ATPase. Sodium potassium ATPase is there. Now, what will this digitalis do is digitalis. Okay, on your myocardial cell, see, on your myocardial cell, apart from sodium potassium ATP, there is also something called as sodium calcium exchanger. Okay, sodium calcium exchanger. Okay, now at the end of the day, I am not going in detail about the pharma. Simple points what you need to know is the digitalis, what it will do. Digitalis inhibits, okay, digitalis, it inhibits the sodium potassium ATPase. Okay, simple. Digitalis inhibits the sodium potassium ATP. Now when sodium potassium ATPase is inhibited, for example, what will happen? Now, do you think sodium is going out? No, sir. Sodium is not going out of the cell. Sodium is retaining in the cell. Try to understand it like this. Now when sodium present inside the cell, when sodium tries to present inside, sodium is not going out. Okay, sodium is retaining in the cell. Sodium is retaining in the cell, and potassium is not coming into the cell. Okay, now what will happen? Can anyone tell me what exactly happens? What will happen, guys? Sodium is not going out. Sodium is not going out. Now the sodium need to be thrown out. The sodium need to be thrown out. So there is something called as sodium calcium exchanger. Okay, there is something called as sodium calcium exchanger. Now all the sodium will try to go via the sodium calcium exchanger. Now sodium is going out. At the same time, more calcium is brought into the cell. More calcium is brought into the cell. Now see, more and more calcium is coming into the cell. Now what do this calcium do? What does this calcium do? Calcium is very important for muscle contraction. I'm not going in detail what exactly happens. There is a lot of mechanism, but in a simple way, for your convenience, I'm explaining. Sir, whenever you are giving digitalis, whenever you give digitalis, so that digitalis will inhibit sodium potassium ATPase. Whenever you inhibit sodium potassium ATPase, at the end of the day, calcium will start to come into the cell. More calcium will be accumulated in the cell. Now this calcium helps in contraction. Okay, this calcium will help in contraction. Now more powerfully the myocardium will contract. Okay, so this action is called as inotropic action. Inotropic action. So digitalis is an inotrope. It increases the power of contraction, not heart rate. It increases the power of contraction of the myocardium. More powerfully, the myocardium is being contracted because of the more availability of the calcium. Okay. Now, having said that, now let's discuss. See, okay, sodium potassium ATPase is completed. Now, what is other examples of primary active transport? See, the first thing is completed. The sodium potassium ATPase, the primary active transport is completed. No doubt. Guys, my question to you, just two important points. Where exactly this proton pump is present? Sodium potassium ATPase is present and it's ubiquitous. It's present in each and every cell. Now, where do this proton pump present? Where do this calcium ATPase present? Can you tell me? I'm trying to integrate as much as with the other clinical subjects. See, right now let's talk about proton pump and also calcium ATP is proton pump and calcium ATPase. Where do this proton pump present? Sir, in your GIT, in your stomach. Okay, in your GIT or in your stomach. Now, see, for example, in your stomach, acid is being produced or not? H plus protons means what? Nothing but acid. In your stomach, acid is being produced or not? Yes, in your stomach, acid is being produced. Now, can you name me what is the cell name? The name of the cell which produces acid called as a parietal cell. Parietal cell. Now, let me show you one parietal cell. Okay, this is one parietal cell. I'm just showing you in an easy way. Let's see. Let's take it as a parietal cell. Now, this parietal cell, it have a pump here. It have a pump called as proton pump. Now, with the help of this proton pump, now this H plus is being secreted into the stomach, into the lumen of the stomach. Okay, so parietal cells with the help of this proton pump, H plus pump, secretes the protons. Now these protons, what will happen? These protons will react with the chloride ions, forms the HCl, acid in your stomach. Now my question, especially for the guys who are studying second year and third year, can you tell me there is a drug which will come and inhibit the proton pump? Okay, if I am suffering with peptic ulcer disease, if I am suffering with gastroesophageal reflux disease, okay, if I am suffering with ulcers in my stomach, what do you have to do? You have to decrease the acid production, right? You have to decrease the acid production. Now, for that, you have to use certain drugs. Now, just name me drugs which will inhibit the proton pumps. Come on, guys. Bruce, Swati, excellent. Pantoprazole, esomeprazole. Okay, Pantap, esomeprazole, omeprazole. Okay. So these are the drugs which will inhibit the proton pumps. And last question here is calcium ATPase. Now, where do this calcium ATPase is present, sir? In the recent exam, this question was asked. Calcium, highest concentration of calcium is present where? In which cell organelle? Can you tell me? Say I am showing you a cell. In the cell, there is nucleus is present. You are having endoplasmic reticulum. Okay, you are having endoplasmic reticulum is present, and also Golgi complex is present. Here is the Golgi complex present. You are having ribosomes, lysosomes, proteosomes. Different structures are there. The highest concentration of calcium is present in endoplasmic reticulum, also called as sarcoplasmic reticulum. Sarcoplasmic reticulum. Okay, now why we are discussing about this here? See, normally here millions and millions, not even millions, like trillions of calcium ions will be present here inside the sarcoplasmic reticulum. There is high concentration of calcium, but still, do you know, on this endoplasmic reticulum there are these transporters present. What are these transporters, sir? So these transporters are example of calcium ATPase. Okay, these are example of calcium ATPase. Now what they are doing, they are all the time moves the calcium into the cell, so not into the cell, into the sarcoplasmic reticulum. Already inside sarcoplasmic reticulum high concentration of calcium is there. In the cell, there is the calcium concentration is less. In the cytoplasm, calcium concentration is less. In the endoplasmic reticulum or the sarcoplasmic reticulum, calcium concentration is more. So calcium is moving from low concentration to high concentration. So this is again is the concentration gradient. So definitely you have to use ATP or not? Definitely you have to use ATP. That's why we are calling it as calcium ATPase. So calcium ATPase is present where, sir? On the sarcoplasmic reticulum. Sarcoplasmic reticulum. Okay. So these are examples of primary active transport which you should know. After primary active transport, now let's talk about secondary active transport. So here again, it's a type of active transport. See, active transport is there. So in secondary active transport, ATP is utilized or not? Just ATP is utilized, no doubt, but directly, no, indirectly. It's an indirect usage of the ATP. Now, so can you give us some examples of secondary active transport? Remember, for your entire life, all co-transporters. Okay, all co-transporters, also called as symporters. Okay, co-transporters or symporters are exchanges, also called as antiporters. Okay, antiporters. They are examples of secondary active transport. Symporters and antiporters are examples of secondary active transport. Now, can you give me some examples of symporters? Can you give me examples of symporters? See, I will explain what exactly secondary active transport, but before, let's see the examples. The co-transporters example are sodium, glucose, co-transport, sodium, iodide, co-transport or symport, and sodium, potassium, 2-chlorine, co-transport. I have given you three examples of co-transport. First of all, what exactly we mean by co-transporter or symporter, sir? Co-transporter or symporter means the transport is happening, two substances are being transported in the same direction. This is important, same direction. Okay, the transport of substances are happening in the same direction. For example, first let's talk about sodium glucose co-transport. Okay, sodium glucose transporter. Now, where exactly the sodium glucose co-transport is happening? Okay, now let's see. This is the proximal convoluted tubule. I am talking about a nephron now. This is a proximal convoluted tubule. Okay, here will be the Bowman's capsule. You all know. Now, this is a proximal convoluted tubular epithelial cell. Okay, now glucose will be filtered or not? Glucose is filtered, sir. Okay, glucose is being filtered. Okay, now first point, where more glucose is present? The glucose concentration is high in the cell when compared to the lumen. More glucose concentration is present in the cell. Now, can this glucose molecule easily move like this? No. Glucose concentration in the lumen is less. Glucose concentration in the proximal convoluted tubular epithelial cell is more. So here diffusion cannot happen. Definitely here diffusion cannot happen, sir. Okay. Now, how to transport this glucose molecule? See, for that, you have to know certain things. On the basolateral membranes here, on the basolateral membranes of this proximal convoluted tubular epithelial cells, PCT epithelial cells, again there are transporters. What are these transporters? Just tell me. So these transporters, just by looking at them, you have to say the name of this transporter is sodium, potassium, ATPase, sodium potassium ATPase. Now tell me what the sodium potassium ATPase is doing. It all the time have only one work. It throws the sodium out. It throws the sodium out, brings the potassium into the cell. It's throwing the sodium out, brings the potassium into the cell. Now it's throwing the sodium out, right? Sodium is being pumped out. So what happened to the sodium inside the cell? The sodium concentration inside the cell is going down by using the ATP. See, by using the ATP, sodium levels are kept low. So ATP is being utilized or not? Yes, sir. ATP is being utilized. So here it is primary active transport, no doubt. Sodium potassium ATP is example of primary active transport, no doubt. Now see, here sodium is there. In the lumen, sodium concentration is more. Inside the cell, sodium concentration is less. So sodium by default, sodium will move from high concentration to low concentration. Okay. So substance is moving from high concentration to low concentration. Now, whenever something is moving from high concentration to low concentration, energy is released. By utilizing this energy, now glucose molecule will move along with sodium into the cell. Okay, see, all this movement is happening because here ATP is utilized, here not here. ATP is not utilized at the place of glucose transport. See, this is the place of ATP utilization, sodium potassium ATPase. Okay, so only if sodium potassium ATPases are being working, if they are working, then only this glucose molecule will be uptaken. So there is indirect use of ATP. Very simple. Sodium potassium ATPases decreases the sodium concentration inside the cell. So sodium will move from the lumen into the cell. Sodium will move from high concentration to low concentration. So when something is moving from high concentration to low concentration, energy will be released. By using that energy, even glucose molecules are moving from low concentration to high concentration. So this type of transport is called as a co-transport. Means sodium is moving as well as glucose is moving in a one single direction. So it is called as symport. Called as a symport. So co-transporters and symporters, they use a direct energy, no, indirect energy. So they are examples of primary active transport or secondary active transport. They are example of secondary active transport. Okay. So all co-transporters are example of secondary active transport. Now you can ask me, sir, what is this transporter? What is this transporter? Which is the transporting sodium as well as glucose? Is it an example of glucose transporter? No. Remember, glucose transporters, glut, they are example of facilitated diffusion. They are examples of facilitated diffusion. Okay, why? Because there in adipocytes, in the skeletal muscles, glucose is moving from high concentration to low concentration. That is a diffusion, guys. They are glucose transporters. These are not just glucose transporters. These are sodium glucose co-transporters. So these are called as SGLT type 2 especially. So sodium glucose co-transporters type 2 present in the proximal convoluted tubule, example of secondary active transport. Okay, now I'm just trying to correlate it with pharma. Okay, pharma. Now imagine I'm the one who is suffering with diabetes mellitus. Now already there is lot of glucose present inside my blood. Already lot of glucose is there in my body. So what you have to do? You have to take out the extra glucose. For example, if you can block the sodium glucose co-transporter, if you can block this sodium glucose transporter type 2, SGLT2 blocker, what happens? Now, do you think this glucose will be reabsorbed? Do you think the glucose is going to enter into the cell? No. What happens? All this glucose is going to lost in the urine. So by this way, you can decrease the blood glucose levels, right? Okay, glucose is being taken out of the body. So blood glucose levels can be controlled. Can you tell me anyone, like Medico, Bruce. Just tell me, can you name me the sodium glucose type 2 transport blocker which is used in treatment of diabetes mellitus? SGLT2 blockers. Cana, excellent. Canagliflozin. Canagliflozin. Dapagliflozin, excellent. Dapagliflozin. Canagliflozin and dapagliflozin. These are the drugs which are sodium glucose transport type 2 blockers. Okay. Now, one question for you guys. One question for you. Simple. Sodium glucose transporters are example of SGLTs are example of simple diffusion, facilitated diffusion, primary active transport, secondary active transport, osmosis. All these are example of secondary active transport. All co-transporters are all antipoters example of secondary active transport. Okay. Now, okay, this one is completed. Sodium glucose co-transporter part is completed. Now, second question, sodium iodide co-transport. Now, where exactly the sodium iodide co-transport happens? In which part of your body sodium iodide co-transport happens? In thyroid gland. Very, very important. Thyroid gland. Okay, you need iodine, right? You need iodine. So, sodium iodine co-transporter is present in the follicular cells of thyroid gland. Okay, now let's take this is one thyroid follicle. Okay, this is one thyroid follicle. Some important MCQs is there. This is the thyroid follicular cell. Now here, please concentrate. Here is a transporter called as sodium iodide symporter. It is NaI. Okay, sodium iodide. Sorry. Actually, we have to call it as like this. Sodium iodide symporter. NIS. Now, what the sodium iodide symporter is doing? It brings the sodium into the cell as well as it brings the iodide into the cell. So, co-transport, 100% co-transporters are example of secondary active transport. Now, this iodide is utilized in the production of T3 and T4. My question for you, especially for the second year guys. Now, I am suffering with a condition called as hyperthyroidism. More T3 is there in my body. Now, do I need further T3, T4 production in my body? Do I need extra T3, T4? Already more T3, T4 is there. I am suffering with hyperthyroidism. I came to you and I am asking you, doctor, I am suffering with hyperthyroidism. Now, just tell me one way. There are many ways. There are many antithyroid drugs. Just tell me one way how to decrease the thyroid hormone production. Can you tell me one way of decreasing the thyroid hormone production? By inhibiting this transporter, sodium iodine symporter, if you block them, automatically iodine uptake is decreased, T34 hormone production is decreased. Now tell me, what are the blockers of sodium iodine symporters? Come on guys, sodium iodine symporter blocker. What are sodium iodine symporter blockers? Anyone? Bruce, come on. Bruce, Ayushi, Swati, Sean Murphy. Come on. Come on, Sean, Ranjan. Not propyl thiouracil. Propyl thiouracil inhibits the thyroid peroxidase enzyme. Propyl thiouracil also inhibits the peripheral conversion. That's a total different mechanism of actions. The thiocyanate and perchlorate. Never forget thiocyanate, thiocyanate, perchlorates. So thiocyanates and perchlorates. Oh my god. Yeah. So thiocyanates and perchlorates are the drugs which will block sodium iodide symporter. Completed. Now last fellow, sodium potassium 2 chloride symporter. Now where do the sodium potassium 2 chloride symporter is present? This is the Bowman's capsule. Here is the proximal convoluted tubule. Now down to the proximal convoluted tubule, you are having descending limb of loop of Henle, hairpin turn, and here is the ascending limb of loop of Henle, distal convoluted tubule and collecting duct. Okay. Now please concentrate. This is the ascending limb of loop of Henle, right? Here is an epithelial cell. Here is an epithelial cell. Now on this epithelial cell, there are these transporters. What is the name of this transporter? See, it is taking sodium into the cell. Sodium. It is taking potassium into the cell. Also, it is taking 2 chloride ions into the cell. Now, is an example of symport or antiport? Definitely, it is an example of symport. So, sodium is moving in a one single direction. potassium and chlorine, sodium, potassium, chlorine, all moving in one single direction. This is an example of co-transport. So definitely all co-transporters are example of secondary active transport. Okay, then no direct utilization of energy here. For the movement of all the substances, there is no direct utilization of substances. If you ask me, sir, see here also, you are having sodium potassium ATPases on the basolateral membrane. Sodium potassium ATPases are present here. Now, the sodium potassium ATPases, what they are doing? All the time they will throw sodium out of the cell. Okay, they will throw the sodium out of the cell. Now, what happened to the sodium in the cell? Sodium levels in the cell decreases. Okay, sodium concentration inside the cell decreases. So what happens? Now sodium will move from the lumen. From the lumen, sodium will move into the cell. Along with the sodium, this potassium and chloride are also being taken into. Okay, this is an example of secondary active transport. Okay, now my question to you, second year guys, can you tell me there is a drug which will block this sodium potassium 2 chlorides importer? The two important questions, sir, for your entire like MBBS duration. You should know these things. Sodium potassium 2 chlorine importer is present where? It's present on the ascending limb of loop of Henle, sir. It's present on ascending limb of loop of Henle. The sodium potassium 2 chlorides importer is an example of secondary active transport. Why? Because it's a co-transport. Now third question, sir. Now what is the drug which will block this sodium potassium 2 chlorides importer? Anyone? Excellent, Bruce. Hey, thiazide diuretics. No, Bruce, someone is saying thiazide diuretics. No, loop diuretics, not thiazide diuretics. Loop diuretics. I see you are true. Loop diuretics. Okay, loop diuretics. Okay, loop diuretics inhibit sodium potassium 2 chloride importer. The example of loop diuretics is furosemide, torsemide, okay, bumetanide, okay, ethacrinic acid. All these are the loop diuretics. Okay, so loop diuretics acts where on the ascending limb of loop of Henle. Loop diuretics blocks which transporter? Sodium potassium 2 chloride importer. Sodium potassium 2 chloride importer. Okay. Now after this, what else you should know? Okay, even we have completed the secondary active transport. The secondary active transport wala business also completed. We have completed the secondary active transport. Now let's discuss the last transport which you need to your exam. The last transport is vesicular transport. Vesicular transport. Okay, let's talk about the vesicular transport. First of all, my question to you, direct question. The vesicular transport, it is an example of active transport or passive transport? Vesicular transport is an example of active transport, passive transport. The simple diffusion, passive. Facilitated diffusion, passive. Primary active transport, secondary active transport, you know, they are active transports. Okay, osmosis. Osmosis is an example of passive transport. My question, vesicular transport, is it an example of active transport or passive transport? Simple, it's an example of active transport. Active transport. Now, what exactly is vesicular transport? Substances are being transported inside the vesicles. Okay, substances transported in vesicular form. Okay, in vesicular form. Now, vesicular transport requires which ions? MCQ requires calcium ions. For the movement of vesicles, if you want to move the vesicles across the cell, you need to have calcium. Very, very important. Vesicular transport requires which ions? Calcium ions. Now, question: how many types of vesicular transport is there, sir? Two types. Two types of vesicular transport is there. First type is called as exocytosis, and second one is endocytosis. Same, vesicular transport is direct area, not too many things were there. 4-5 MCQs are there. 4-5 MCQs are there, and I hope you already know this. Now, my questions to you: what exactly is exocytosis? What exactly is exocytosis? For example, let me show you a cell. This cell. Now, here is a substance. For example, here are certain substances which are present inside a vesicle. Now this vesicle, with the help of calcium ions, now this vesicle is moving where? It's moving towards the membrane. Okay, it's moving towards the membrane, and this vesicle, it's moving. It's coming towards the cell membrane. Now this vesicle, it will go and fuse with the cell membrane. It will go and fuse with the cell membrane. It releases the substance. It releases the substances outside. So this movement is called as exocytosis. Exocytosis. So from inside to outside, the movement of a substance through a vesicle from inside to outside is called as a exocytosis. Now my question is, for the exocytosis, you need to have a special proteins. Okay, you need to have special proteins for the process of exocytosis. Can you name me? Exocytosis requires which protein? Direct. It requires cladherins, it requires megalins, it require caveolins, it requires synaptobrevins. The name of the proteins which are required for the exocytosis. Yes, exocytosis is called as cell vomiting. Excellent. Okay, there is one more name if you can tell me. Exocytosis is called as cell vomiting. It is also called as cell vomiting. Cell is vomiting out, spitting out. It's also called as emiocytosis. Okay, EMEIO, emiocytosis. Now the proteins which are required for exocytosis are called as MCQ snare proteins. S-N-A-R-E. Snare. Snare proteins are involved in exocytosis. Okay, now here I want you to know two important snare proteins. Can you tell me the two important snare proteins which are involved in neurotransmission? The two important snare proteins which are involved in neurotransmission. Anyone who is really good with the neuronal transport? See, this is the axon. This is axon. This is a nerve terminal. This is a nerve terminal. This is the presynaptic membrane. Presynaptic membrane. Here is the postsynaptic membrane. Okay. Now my question, see, here is a vesicle. What is present inside the vesicle? Neurotransmitter. Neurotransmitter is there. Okay, neurotransmitter is there. My question is, see, now this is vesicle, right? Vesicle. Now this vesicle is coming. Now on the surface of this vesicle, there are snare proteins present. Now what is this snare protein called as, sir? Very, very important. You should know this for your entire life. So this snare protein is called as synaptobrevin. Synaptobrevin, sir. And there is one more protein present in the presynaptic membrane. This protein is called as syntaxin. Okay, synaptobrevin and syntaxin is there. Now this vesicle will move like this, and this vesicle, what will happen? Now this vesicle with the neurotransmitter, now the synaptobrevin is going to come and attach to the syntaxin. Synaptobrevin, syntaxin are going to fuse. Now then only this vesicular membrane, it will fuse with the presynaptic membrane and neurotransmitter is released into the synapse. Okay, so what I am saying, this is cell vomiting, right? It's exocytosis, right? So what is the important protein involved here? Synaptobrevin, syntaxin, example of snare proteins. Now the clinical importance here, sir, the clinical importance. Now whenever you got infection with an organism called as Clostridium botulinum, okay, whenever you got an infection with the Clostridium botulinum. Now my question for you, especially for the exam going batches, 4th year, 5th year students, even 2nd year students who have studied the microbiology. So Clostridium botulinum will enter into your body. It will enter into your body whenever you eat which foods? Clostridium botulinum enters into your body whenever you eat which kind of foods? Contaminated milk and meat products. So contaminated milk and meat products will bring you Staph aureus into your body. Then eating what kind of foods will bring Clostridium botulinum into your body? Excellent, Saad. Excellent, Bruce. Canned foods. Okay, in supermarket you will be having this canned food, right? So canned foods. So food stored in the can. So whenever you take this canned food, in that canned foods, Clostridium botulinum spores may be present, or even Clostridium botulinum is present. Whenever you take this Clostridium botulinum spores, Now those spores will come into your body. Now this Clostridium botulinum, it will produce a toxin called as botulin. It will produce a toxin called as botulin. Now what the hell does this botulin will do, sir? Now this botulin, it will come and it will break down the synaptobrevin. Synaptobrevin is being broken down. Now there is no synaptobrevin, syntexin, no synaptobrevin, syntexin, they cannot meet. Now when they cannot meet, neurotransmitter, do you think the neurotransmitter is going to be released? There is no release of neurotransmitter. Acetylcholine, it's a neurotransmitter, right? Acetylcholine is a neurotransmitter. Now acetylcholine is not being released, sir. Acetylcholine is not being released. Now acetylcholine is very important for the muscle contraction. Okay, nerve is coming like this at the neuromuscular junction. At the neuromuscular junction, acetylcholine should have to be released from the vesicles. No, there is no acetylcholine. Whenever there is no acetylcholine, do you think the muscles will be contracting? Muscles are not contracting, sir. So the patient is going to have what? The patient is going to have paralysis. So why I am discussing all this, especially which type of paralysis? Flaccid, flaccid descending. So especially in your exams, if you see this word, descending flaccid paralysis, descending flaccid paralysis. Okay, because paralysis is of two types: descending paralysis, ascending paralysis. Ascending paralysis is seen with Guillain-Barré syndrome, as a descending paralysis is seen with botulinum toxicity. The patient is going to have flaccid paralysis. Flaccid means he cannot contract his muscles. All his body is like this, flaccid. Okay, no contraction, flaccid. Okay, muscles are not having any tone; they are just flaccid. So, descending flaccid paralysis is seen with eating contaminated food with the Clostridium botulinum. Okay, so this is one type of transport. That's the exocytosis, where you need to have snare proteins like synaptobrevin and syntaxin. And for endocytosis, okay, for endocytosis, endocytosis, you again need to have different type of proteins. You again need to have a different type of proteins. What exactly is endocytosis? Imagine this is a cell, this is a cell. Outside, okay, outside there are certain substances present. Okay, there are certain substances present. Now what will happen? Now cell membrane is going to become like this. Now cell membrane will uptake the substances which are present out of the cell. So from movement is happening from out to in. Okay, so from out to in, so in the form of a vesicle. Okay, now it forms a vesicle and this vesicle is going to come into the cell. Now this type of movement is called as endocytosis. But direct question, endocytosis require which type of proteins? Clatherins, clatherins and megalin. Okay, so see, PDF will be shared to you. I asked the management to share the yesterday's class PDF and today's class PDF in the Telegram. Okay, after the class I'm going to share yesterday's class and today's class PDF. You don't worry about the PDF. So endocytosis is going to be happening with the help of proteins like clathrin and megalin. But important MCQ is what I want you to know is, sir, endocytosis is again of two types. Very simple. Just names you have to know. Endocytosis means of two types. One is phagocytosis and pinocytosis. Pinocytosis. Pinocytosis is also called as cell drinking. Okay, for example, here some fluid is there. Now that fluid is taken into the cell in the form of a vesicle. So that is called as a pinocytosis. What is phagocytosis? Here outside the cell, especially outside the macrophage. Okay, let me show you here. See, so this is a macrophage. Okay, this is a macrophage. Now outside the macrophage, you are having a bacteria. Bacteria is present. Okay, let's take this as a bacteria. Bacteria is present. Now what our macrophage will do? Our macrophage will produce, will protrude our cell membrane like this. We will protrude our cell membrane like this. These are called as the these are called as the pseudopods. Okay, now these pseudopods will completely surround the bacteria and will uptake this bacteria, will take this bacteria into the cell. The bacteria is taken into the cell inside a vesicle. Okay, this is a vesicle. Okay, let me show you. Okay, let's take, this is a vesicle and inside the vesicle there is this bacteria. Now this type of uptaking, engulfing a bacteria by a macrophage into the cell, this moment is called as phagocytosis. Phagocytosis. Now you all know, second year students, you have studied about the pathology, the cell topic. Now this phagosome inside here, what is there? This is a phagosome. Now this phagosome is going to be fused with what? See, inside the macrophages you are having lysosomes. Okay, inside the macrophage you are having a lysosome. Here is a phagosome. So phagosome is going to be fused with the lysosome. What happens now? Phagolysosomal fusion is happening, sir. When phagolysosomal fusion happens, all the hydrolytic enzymes, acidic enzymes which are present in the lysosome are going to be released into the phagosome. So the bacteria will be destroyed. Bacteria will be killed. Now why I am saying this is because, see, the topic is very simple. Phagocytosis means, now the antigen presenting cells are the macrophages. They will produce the pseudopods, take the bacteria into the cell inside the vesicle. So this movement is called as a phagocytosis. After this, the phagosome which contains the bacteria is going to be fused with the lysosome. This is called as a phagolysosomal fusion. Once the phagolysosomal fusion happens, the lysosomal contents will act on the bacteria and kill the bacteria. Now why I am saying all this is because, see, there is a disease where phagolysosomal fusion is defective, which means the phagosome is not going to be fused with the lysosome. So now do you think in that condition bacteria will be killed? Now whenever you got infection, bacteria is there inside your body. All the macrophages should have to go and destroy that bacteria, right? A macrophage is eating the bacteria, phagocytosing the bacteria, but lysosome is not coming and fusing because of a mutation called as list gene mutation. List gene. List gene is mutated. Very, very important. Because of list gene mutation, phagolysosomal fusion is defective, so that this person is going to have recurrent infections. Why? Because the bacteria is not getting killed. Can you tell me what is the name of this disease? Can you tell me what is the name of this disease? Come on, guys. The name of this disease is called as Chediak Higashi syndrome. Okay, so in Chediak Higashi syndrome, the patient is going to have defective phagolysosomal fusion due to list gene mutation. Okay, so with this, we have completed all the different types of cellular transport. Okay, I hope I thought to complete even the cellular cell organelles also, but it's not possible. Why? Because the concept of this cellular transport is little big and it's highly conceptual, right? I tried to integrate with as many clinicals as possible, as many the pharma points as possible. So I hope this will be enough for today's class, and the next physiology class will be doing cell organelles and some other topic also, like cellular messengers. So in the next class we will do cell organelles and cellular messengers. If you have any doubt, just put it in the chat box. Just keep your doubts in the chat box and the PDF will be shared in the Telegram group. Okay, guys, do you have any questions? Class is helpful? See, when is the next class? The entire schedule of the classes will be updated in the Telegram group. I think by this time the management have already posted there. See, right now, according to the schedule, we are going to teach the general physiology. First, let's complete the general physiology. Later on, we will do the systemic physiology. Okay? Okay, guys. See you in our next class. Tomorrow, we will be doing the patho. Tomorrow, first chapter, general pathology. I'm going to do the first chapter of general pathology. Okay? Simultaneously, I will be doing physio as well as patho. Tomorrow, patho. Okay, guys. Bye-bye.